So in the first slide, you can see all the immune cell or blood cells are produced from a single originator pluripotent stem cell called hematopoietic pluripotent stem cell. This production of this all blood cells are called hematopoiesis. So from the hematopoietic stem cell, common lymphoid progenitor can develop and common myeloid progenitor cell can be developed. From that common lymphoid progenitor, B cell, T cell, NK cell and ILC means innate lymphoid cell, immature dendritic cell can be developed. So this occurs in the primary lymphoid organ, but these cells can come out into secondary lymphoid organ like lymph node can become activated, then it called effector cells. B cells in effector condition called plasma cells, and immature dendritic cell can become mature dendritic cell. From the common myeloid progenitor, granulocyte and macrophage progenitor can develop. Granulocytes are neutrophil, eosinophil and basophil. They are also called polymorphonuclear because they contain many lobes of nucleus. Precursor of macrophage is called monocyte. Immature macrophages are called monocyte, and this is the neutrophil. They contain multi-lobe nucleus, so they are called polymorphonuclear leukocyte. Neutrophil constitute the majority, like 50% to 70% of the circulating WBC. And one interesting fact is that a healthy individual, a human healthy individual, can produce more than 1 into 10 to the power 11. That means 1000 crore of neutrophil per day. And another function performed by neutrophil is phagocytosis and secrete some antimicrobial substance like lysozyme, cathelicidins and defensins. Also they can produce some enzyme called collagenase, elastase, etc. This neutrophil contain some granules, and these granules actually contain this antimicrobial substance. Another important function of neutrophil, during infection, some neutrophils undergo a unique death in which their nuclear chromatin do not degrade. What happens during apoptosis? Nuclear chromatin degraded. During this procedure of death, neutrophils' nuclear chromatin come out and form a fibril-like structure called NET, neutrophil extracellular traps, that can trap extracellular pathogen like bacteria and fungi. These rod-like structure are actually bacteria. These are basophil and eosinophil. These are all neutrophil, eosinophil, basophil are granulocyte. They contain granules. You can see they all contain granules. And this basophil and eosinophil perform more or less same functions. They develop immunity against helminth and produce allergic symptoms. This basophil and eosinophil actually express receptor called FcR that is specific to epsilon to produce allergic symptom, to develop immunity against helminth. One important antibody is IgE. This is only responsible antibody that actually develop immunity against helminth. Eosinophil, basophil and also mast cell express high affinity Fc receptor for IgE antibody. Fc receptor means fragment of crystallizable receptor. So this receptor can bind with the Fc gene of antibody. Antibody can coat a helminth, and that antibody can be recognized by eosinophil, basophil and mast cells, and after binding it with this Fc receptor or epsilon receptor, degranulation process starts, and these granules of basophil, eosinophil, mast cells are contain vasoactive mediator like histamine, heparin and leukotrienes, prostaglandins and other substances. So they can degranulate their substance that are present in the granule and can kill the helminth. Also, this granule containing proteins or chemicals can form allergic symptoms like asthma, hay fever, hives. Now you can see the mast cell. This is a mast cell, and the mast cell contains granules. Here it is showing granules. And how a mast cell works? Here it is showing that T cell or subset of Th cell called Th2 cell actually can activate a B cell in such a way that the B cell can produce IgE molecules, because Th2 cell secrete a chemical called IL-4 that helps to produce the B cell IgE antibody. A B cell can bind with this antigen or allergen that can be represented by MHC2 molecule to the T cell, and the Th2 cell actually respond this signaling by secreting IL-4. After becoming plasma cells, the B cell produced IgE antibody that are specific to that allergen. IgE molecule can bind with this allergen. When an antibody membrane condition can bind with a particular antigen upon secreted form or soluble form, they also can recognize the same antigen because their variable region do not change. So this antibody IgE molecules can bind with particular receptor called epsilon receptor, FC receptor for IgE. These are high affinity IgE receptor, and these mast cells are called sensitized mast cells. If there is another attack of allergen, the allergen can cross-link the antibody and degranulation process starts. So all the chemicals that are contained in the granule are released. So they can produce, these are vasoactive amines, so they can produce many physiological symptoms like smooth muscle contraction, blood vessels means small blood vessels, the vascular permeability increase, mucus secretion, and other functions like sensory nerve endings activation, attraction of eosinophil. In the next slide, you can see the function of macrophage. This is actually a macrophage that is engulfing bacteria or phagocytosing a bacteria. The major function of macrophage that it is a professional APC, antigen presenting cell, express MHC class 2 molecule, and it can activate effector T cells, not the naive T cells, but effector T cells, can phagocyte as it is showing in this picture, and also very different role can be played by a type of macrophage that is tissue repair. So there are actually two types of macrophage. One is called inflammatory and another is called patrolling. Ultimate function of the macrophage, that macrophage express toll-like receptor. I have already told it in my previous class that toll-like receptor, pattern recognition receptor. They can recognize some PAMP. PAMP means pathogen associated molecular pattern that are present on the microbes, signature molecular microbe. And these toll-like receptor are the recognition receptor. And also the macrophage express cytokine receptor. And one cytokine is very important for the activation of macrophage is called IFN gamma. When there is IFN gamma, the macrophage or resting macrophage can be activated. During the process of complement pathway, some fragments can be released, and these fragments attaching with the microbe can bind with complement receptor and activate a process. So you can see all these receptors actually activate some cytokines production, phagocyte oxidase activation so that ROS can be produced, reactive oxygen species. This reactive oxygen species actually helps to kill the phagocytose bacteria or pathogen, production of nitric oxide. The ultimate result is killing of microbes, and by secreting some cytokines it actually induces inflammation so that other cells can recruit in that area. So inflammation is the red signal of our body. Here you can see generation of macrophage. Macrophage can be generated from fetal hematopoietic organs like yolk sac or liver. When the embryo matures, it comes from hematopoietic stem cell. This macrophage, when it is immature condition, is called monocyte, can go different organ like brain, liver, lungs, spleen or bone. Depending on the tissue, its names differs. Like in the brain, the macrophage is actually named microglial cells. In the liver, it is called Kupffer cells. In the bone, it is called osteoclast cell. In the lung, it is called alveolar macrophage. And in the spleen, it is called sinusoidal macrophage. Now you can see the major types of macrophage. One is called classically activated macrophage M1 and another is alternatively activated macrophage called M2. The first M1 is called inflammatory macrophage that can produce or actually induce inflammation. And the alternatively activated macrophage function opposite to the M1. These are called patrolling macrophage. When the antigen or the pathogen are eliminated, this macrophage actually perform their function. They function totally different. As you can see, this from this monocyte, both cell can be produced, but the responsible cytokines are different. In case of M1, the IFN gamma is responsible. TLR ligands and microbial activations are important for the transformation of monocyte into M1, and this M1 cell actually can induce inflammation. So you can see microbial actions, phagocytosis and killing of many bacteria and fungi and inflammation by producing IL-1, IL-12 and IL-23 and chemokines. Type 1 interferon is produced by Th1 cell. So the production of macrophage M1 is induced by Th1 cell. But this type of cytokine like IL-13 and IL-4 are produced by Th2 subtype of Th cell, Th2. So this is Th1 and this is Th2. That's called M2. These cells can produce IL-10 and TGF-beta, that these two cytokine are anti-inflammatory cytokine. So this cell actually repair tissue and this cell actually kill the pathogen. So the two macrophages actually perform the opposite functions.